have Scott Zeger. So you know, Scott came with Kong Yi in 1982. Senior Associate Dean for Academic Affairs from 91 to 96 at the school. He was Chair of Biostatistics from 96 to 2008. Became Vice Provost from 2008 to 2013 and served as a provost for about, the actual provost for about 9 months. Right now he's Director of the Hopkins in Health Initiative. He started the journal Biostatistics. He's had so many awards, honorary degree from University of Lancaster School of Medicine, Marvin Zellin Leadership Award from Harvard, Bradford Hill Award from the Royal Statistical Society, member of the National Academy of Sciences, Institute of Medicine, Golden Apple Award, Fellow of the American Association of Advancement of Science, Spiegelman Award, Snedeker Award, on and on and on. So, you know, this was really, really depressing going through, you know. And he's got a Google Scholar page, and so I looked up and said, well, come on. 12,137 citations for the Liang and Zeger paper in 1986. Unbelievable. So, welcome, Scott.
Well, thank you much, Dan. My mother would appreciate it. So it's been an interesting week, actually, getting ready for being with you, with Chuck today. And I actually started out imagining this would go one way. And just in thinking through the things I wanted to say, you know, and really Chuck's influence on my own career, I found myself shifting more and more to more personal thoughts. And so anyway, I apologize for that if it gets too much that way. But before I start, I want to say that I want to thank Karen and Mei Chen for allowing me to be a part of this part of the session. We weren't sure how many of the melon heads would be able to get here, and so when so many came, we thought we would organize it this way. And so I'm appreciative for the chance to speak. I really want to say that when I came to Hopkins, and I'll talk a little bit about that, I became a part of a community you've heard about, and really anything that has that I have been able to, any small things I've been able to contribute as a result of the collaborations with all of you, and they were all made possible by Johns Hopkins, by Johns Hopkins School of Public Health, and really by Chuck's leadership that created the environment in which really magical things have happened, and it's been a privilege to be a part of it then and now. So I was going to talk about some interesting research projects that I'm fortunate to work with some postdocs on, but as I said, I started thinking about this. I really realized a number of places, just the way Steve said just now, where Chuck influenced me in a way I probably hadn't realized until I was thinking about giving the talk. So. I'm going to tell you when I met Chuck. He may not know about this event anymore, but I was well. I'll tell you about it. And one of the things I learned from that was a meeting together in a big group of people, and he made a presentation about some of his work, and I realized looking back that was really a turning point in my career. I apologize for this numbering. I tell you a little bit about oysters. And again, I had been, you know, on the edge with oysters for quite a while, and I'll tell you about how Chuck made a difference there. I was not a particularly successful junior faculty member for quite a while when I first got here, and I want to talk about the things that Chuck did to get me over the hump, and I think probably most of them was by getting Steve, Ron, and Kong Yi to push me in the right direction, and then these were in order at one point, but I have an apple, and there may be some significance in the numbering that I'm yet to be aware of. But I've always watched him enjoy companionship and enjoy mentoring. So those are the things I want to talk about. There's only, you can see only two things there, so it shouldn't take too long. So let me start with the first contribution Chuck made to my career. When I got out of college, I couldn't find a job. I had learned how to program an IBM 360 computer as an undergraduate, and I eventually got a job at a place called the Academy of Natural Sciences in Philadelphia. And that picture is a picture of Ruth Patrick, who is a fellow of the Academy of Natural Sciences and was a limnologist. She studied those things on the right, plankton, a kind of plankton called diatoms, and they are special because they leave silicon shells, and so you can take cores and look at the history of the diatom population for tens of thousands of years in the past, and there's also tens of thousands of species that have different tolerances, so you can learn a lot about the environment from their history. And the Academy of Natural Sciences had a business at the time. Ruth studied diatoms, but they had a business to put people like me. I got a job there as a new graduate from in Philadelphia, and got a job. I think I'm that guy up on the left there. You can't see him. He's sort of down in the bushes because I was not quite ready to go into the streams. But anyway, we would catch fish and sample diatoms, and we'd talk about the pollution effects of usually industry on the Chesapeake and Delaware bays. Okay, so I was doing that job. It was great, summers on the bay, and I was enjoying it. And I got to go to a meeting in Annapolis, and it was a meeting. I was too low to go, but they maybe needed a drive or something, and I went with some people. And there was a bunch of the bigwigs from the state of Maryland Environment Administration and others, scientists who were thinking about the quality of water in the Chesapeake Bay. And Chuck Rohde was there representing Johns Hopkins University. And I listened to a bunch of policy people talking about stuff that didn't really make a lot of sense. There was a lot of airy-fairy kind of stuff. And then Chuck Rohde actually started talking about how you should be sampling to be able to monitor the bay. And he actually was talking about what I subsequently found out was this paper, which he had published in Biometrics, and he was talking about being able to actually measure the quality of the bay rather than talking about it in the abstract. And as a young person who was driving somebody to the meeting, I thought, now that's somebody who is worth emulating. And I think we probably shook hands at that meeting, and I don't know if there was ever a reconnection in your mind, but as I was thinking about, why did I go to graduate school? Because I was, just working. Why did I go to what was it about my job that got me interested? And I do remember that moment as being very influential in my seeing the capacity for rational thought and science and sort of quantitative science being a basis for solving some of the problems that were being discussed. So, and I just want to say that what Chuck did, which was different than everybody else, is he framed the problem precisely, and he abstracted a quantitative statistical issue from the substantive one. At about that same time, maybe a few years earlier, I lived in Philadelphia, and this is the Schuylkill River running to the north downtown, and right there where the arrow is, some of you can see it maybe, is a beautiful little park. And here's a view from you can see the boathouses, and the park is just beyond the boathouses. And I was in Philadelphia one day, and it was a beautiful day, and I went out to that park, and I was laying there on the grass watching the rowers, and I took a book with me, which was this book, A Movable Feast. And I didn't have any expectations about it, although as I was thinking about this, Chuck does have some of Ernest Hemingway's inclinations, let me say, for good food and wine. And this is a book about Hemingway wrote about his years in Paris. And I'm reading along, and I'm from a small town in upstate New York. We never saw oysters in that town. But I once was taken by my parents to New York City, and I remember there was a famous oyster bar, and my mother and father stopped. I was maybe 8 or 10 years old. I was standing next to them, and they ate some oysters, and I thought it was a grotesque thing, but it did sort of get into my mind, And anyway, I'm reading on the beach there next to the Schuylkill River, and I read this. As I ate the oysters with their strong taste of the sea and their faint metallic taste that the cold white wine washed away, leaving only the sea taste in the succulent texture. And as I drank their cold liquid from each shell and washed it down, the crisp taste of the wine I lost the empty feeling and began to feel happy and to make plans. So it was a beautiful moment. I thought to myself, maybe I should try oysters, you know. But then I thought to myself, nah, you know, until I came to interview at Johns Hopkins. So Chuck also picked me up at the airport, and Chuck in Sevilla, and that meant something special. I had been here maybe a month before to interview at what was then, I think, Math Sciences. And I wasn't treated very nicely, I would say. It was not particularly welcoming. The only welcoming thing about that visit was that I remember Ron and Steve, maybe Hendricks, coming to the talk and introducing themselves with a lot of enthusiasm, unlike what I was seeing in the rest of the faculty, and thinking to myself, boy, I'm lucky to have an interview at the Department of Biostatistics. Anyway, I had a wonderful interview, and I felt totally at home. And then after the interview was over, I was taken to Chuck and Sevilla's home for an oyster party. And I'd never tasted an oyster. I had read that book, but there they were, sitting there. And I can remember thinking to myself, you know, I'm gonna have to eat these oysters. And I remember looking, and there was Steve and Ron and Hendricks. They were at this party, and they were sort of smirking off at this, thinking to themselves like, this is the trial by fire. Let's see what he's made of. Yeah. So, sorry. Oh, anyway, make a long story. Probably this will cancel the whole thing here. Right? So try again. To make a long story short, I did eat them, and I enjoyed them, and I have gone back and read that Hemingway passage a number of times. And now when I go to Paris, I sit down and have oysters and white wine and enjoy it. And really, I put that, Chuck, at your through your influence. The third thing I want to talk about is being patient with junior faculty. And so this is the top of my CV. And if you notice, the key was that I earned my PhD in 1982. And if you read a little bit further along, you'll start to see papers published. And if you look carefully, what you'll notice is that I didn't write any papers, basically, for about 3 years. And the first, I had tried to get some papers from my thesis, and I was working on them, and eventually I did publish a couple, but it was like a dry spell. And you'll notice that the first paper I published, which was with Steve and Kung Yi, and they were working on this problem, and kindly invited me to come to some meetings, and I listened in. I didn't know what they were talking about. I had absolutely no idea about most of what they were doing. And at some point, they said, well, we should write this up. And I'm pretty certain that Chuck and Steve were on the same wavelength. And Steve had been there. He knew he fished with Chuck. There were things that, and I'm sure Chuck said to Steve, why don't you see if you can take that guy under your hand? It's been a while. I'm not sure he's got it in him. So maybe you and Kung Yi could sort of get him involved, and they did. And we got that paper published, and they told me that I should be first author, but I didn't really get that. But I didn't have any papers, and so I said, oh, okay. And but really, Chuck never once said to me, what are you doing? It's like, what are you working on? He just in small ways got me involved in collaborations, and it made a big difference. And I was thinking about what was it like to be here when I first got here? What was I doing? What was I working on? And I went back and thought about, well, what did I know about when I got here? And I put those things on the left. I knew a lot about so-called exploratory data analysis, GH technology, 3R SSH smoothers, and all that sort of stuff. I knew a lot about frequency domain time series analysis. In fact, I taught a course here. My first course I taught, I started out with like 4 or 5 people, and I ended up only with myself and Dave Wilson, who was a professor at the University of Maryland, because I was teaching about frequency domain time series analysis, and there were no time series data here, they certainly didn't care about that. And anyway, I knew about stuff. And the other thing I remember is how John Tukey used to be just, he was so disrespectful of any data that wasn't like continuous, and a real measurement. He thought binary data was like inferior, and censoring. Well, why didn't they just measure it? It's like, so. Anyway, and then I got here, and the things you need to know about are all the things that they treated with some disrespect where I came from. And I had never heard about a lot of the stuff that ultimately really has been important in biostatistics. So there was a time that was required to get up to speed. And I think about the department. And so I was thinking about some of the really influential people in the department. And I remember Karen, when she first came here, had written papers about so-called source apportionment, and which was a sort of a specific area with applications in physical science and air pollution, and she knew a lot about latent variables and took that and really became a driver of our understanding of human frailty and health, and a lot of the ideas that she had developed carried over, but it took a while to build that connection, and then. The best example of this was Rafael Irizarry, who, if you remember, came and gave us a lecture about castrato expression, and I won't go into the details, but it was really about mathematics frequency domain models for music, and ultimately moving from the expression of the castrato voice to gene expression, and made a big influence there. And I see Brian Caffo is not here, but now you may think I'm being disrespectful, but this is actually the picture on his website. I didn't. But Brian came here talking about technical issues around p-values. I remember at the faculty meeting afterwards, Chuck assured us that he would be a fantastic faculty member here. After everybody just sort of ripped to shreds everything he had talked about. And sure enough, he's now talking, I had a conversation with Marisa, he's talking about inference-free data science. So people do transition over time, and it does take time. So I think Chuck understood that, always understood that, and really made time for us. So the third thing I learned from Chuck was about enjoying companionship. He always seems to be able to enjoy companionship at amazing places like this one. And we tend to go to Dundalk, but I actually knew how important family was even before I came here, and I thank my own family for all the love and companionship they've given me. But really, the more important question is, who's a man's best friend? And Chuck always knew that, and he taught that to other faculty, and Ron followed in his footsteps, and it took us a long time, but we eventually got a dog too, and so Karen everybody got dogs, and it really has been transformational for our family, and so Chuck, I thank you for that. We don't quite have the same calibers of dog, but anyway. And then I want to talk a little bit about mentoring, and I'm just going to give a couple of examples of some scientific studies that I'm having the privilege of mentoring two young people in the area of so-called big data. And so this is just a digression from personal part to try to give some scientific legitimacy to this last bit of the talk. And really, we're in a new age now where what we measure about people is much more complex than has been the tradition. And it's very, very exciting because we now are measuring things that are fundamental to the mechanisms of health and public health, and these are just meant to be some of the kinds of data that you get. And so really the question is, how can we use these data the way that we've been using data all along? And my own interest of late has been, maybe I'm getting ready to be a patient, but it's really been from the perspective of the individual patient and the decision that patients make with their doctors. And so I use this example just to try to frame people to think about what it's like for an individual. You go to see a doctor, and the doctor has a little bit of data about you, simple data, like in this case that you're 50, not me, but that the person might be 50. that they don't have any family history of this particular disease that we're talking about, but that they had tested positive in this screening test. So you, the doctor, and the patient are trying to understand, well, what is the state of this patient? That's the question. And you have that limited amount of data. So what do? So what we have always done is we then think about a population of people who are in the same situation, and we look at the positive predicted value of that test, which is very low, and so we now can interpret the finding. And so what's different now is that we want to be able to do that with more complex data, right? And so so the whole notion is to create computational analogs of that 2x2 table, but where the data are much more complex. And ultimately what we'd like to do is to be able to make reference populations that are ever more precise in how they match the person at hand, precise in terms of the genomic and epigenomic and image kinds of data that we can now measure for people. So the goal really is to try to reference a person to an ever more narrowly defined group of people for whom the outcomes are more informative. So that's the problem. I'm just going to jump over all the stuff and go to two examples. So what we're trying to do is to do that 2x2 table, but now in more complex situations. And I'm just going to show you two examples, one in which the goal is to say what is causing a child's pneumonia, and this also is, like Steve's project, is work that's generously funded by the Gates Foundation, and a second example where we're trying to say what's a person's prostate cancer state. So here's a child with pneumonia. Pneumonia is a clinical condition. It involves fever and cough, etc. But it's a clinical condition that's caused by many different diseases, all infections of the lung, and you can't be the Gates Foundation and dramatically reduce the rates of pneumonia, which is the leading cause of child death in the world, unless you know what the disease is, right? So sometimes they're viral diseases, sometimes they're bacterial diseases, can be funguses. So you have to figure out what's in the child's lung, and unfortunately you can't observe what's in the lung. You can't stick a needle into the lung, for example, and make measurements, and they have done a little bit of that, although it's not even that informative. So here's a model of the problem. You want to know what's in the lung, and what you get to do are measure the periphery. So you have blood culture data, possibly. You have data from the nasal pharyngeal cavity, from the sputum, and the goal of this project is to take data from those peripheral sites and to say what are in the child's lungs. And so this is very much like, this is a latent variable problem, where the latent variable of interest is what's in the lung, and then you have, given the latent variable, you have measurements from the periphery. And so Jianke Wu and others on the team have devised some very clever ways to say, in a population of people, what's likely to be the cause of infections in those lungs. And what we get on the left there is the data we observe. So we have 5,000 cases and 5,000 controls. And you see the blue lines, there's a blue line, the top dot on the left is the control rate, and it's connected to another blue dot that's the case rate in the nasal pharyngeal cavity. So we have, for all the major pathogens, the difference between cases and otherwise matched controls. Sometimes when it's slanted down, that means there's more in the cases than the controls. When it isn't, it means when it's straight up and down, it means you see at the same rate in the cases and the controls. And then we have blood culture data where with low sensitivity we can detect bacteria in the blood. And if there is a bacterium infection in the blood, we know that it's come from the lungs, so we get better data there. And actually that data helps to inform the bacteria data that's from the nose. It helps us train up the data in the nose. We do know something about the sensitivities of these measurements from outside sources, from vaccine trials. And so we actually have prior information about the sensitivities of the two kinds of measurements, which are shown as distributions, as sort of box plots on their sides. And then what you do is you combine the rates that you observe in the nasal pharyngeal cavity with the rates you observe in the blood, and with prior knowledge about the sensitivities and distributions for those, and you end up with posterior distributions for the rates of all the major pathogens. And you also then can say for a particular child, so look at the child as the top row, the third one over from the left. That's a child that had RSV positive in their nose. You don't see that in the controls very often. It's a very good indicator that they have RSV, and so the posterior distribution for that one child is, it's definitely RSV, right? That child also, if you see the top there, it says 100. The one on the far right is RSV. The first one is, I think, Haemophilus influenza. Turns out you see that just as often in controls as in cases. When you see it in the nose, it really doesn't mean anything. So we end up assigning that kid with almost 100% probability to RSV. So for some kids, they get assigned with high certainty to the various pathogens. For others, we just don't know about them. And so it's an example of trying to take more complex data, here many measurements from the periphery, to say what's the state of this child's health, very much like we did with the 2x2 table. And then the last quick example is in prostate cancer. It turns out that, well, at Hopkins there's about 1,300 men that are in a group called the Carter Active Surveillance Group, and Bal Carter is a very well-renowned cancer surgeon, but who has decided that we do too much prostate cancer surgery, and that for many men the best choice is to just actively be in active surveillance and to watch over time. And so, and that's because they've now, observing a large cohort of people, see that about half of the people who used to get surgeries have non-lethal tumors, have tumors that are really indolent and would have been best left in place. The surgery has very significant side effects. And so the problem is like the 2x2 table. This is what you're given for the individual. Instead of positive on a test, you're given a time series or sequence of measurements of a prostate-specific antigen, and also down at the bottom, these places where they come and have biopsies. And these are all men who had equivocal biopsies, Gleason score 6s, where it's that cohort where we're really not sure that the cancer is indolent or not. And so this is a man who had 4 such biopsies, and so this is the data. And this is like the positive test. We want to refer this to a population of people. So we have 1,300 men represented there on the right. We also have information about the measurement error from following these men forward. And so what we do is we do very much like we did for the child's pneumonia case. This is the PSA data and after the first biopsy for that man I showed you. And we would say that he has a 52% chance of having a lethal tumor, and we can predict where his PSA is headed, and also that if he had another biopsy, what the probability of reclassification would be. And they use this now, hopefully, to decide when to have the next biopsy. This is historical data, so we actually had 10 more years. You could see his PSA took a turn for the better. Big is bad in PSA scores. And our predictions didn't prove out so well, but we now have new predictions, as always the case. And he also had 3 more equivocal biopsies. And so now we feel that his probability of being a lethal tumor is down from 52 back then to 5 years later, 9%. And so we're going to keep following. We're not going to do anything. And sure enough, it keeps on the way it was going. And after 10 years, he's still not had any intervention, and he's at a 5% risk of having a lethal tumor. So these are meant to be examples of using statistical ideas to try to reference an individual about whom we have some data, maybe more complex kind of data, against a population of people, and to be able to do better by them than we would have if we just sort of thought about the problem. So I want to close. That was supposed to be about big data. I think I'll just skip this and say once again, so Chuck, thank you very much for creating the environment at Johns Hopkins, for teaching me, and I think many others, how to abstract important questions from real problems, how to eat oysters. That was tied for #1, Chuck. And thank you for being patient with this junior faculty member and allowing me to get beyond those first 3 years. And as all of you know, it is a time of uncertainty, and you're not really sure whether you're going to get beyond it or not. And you were patient with all of us, and hopefully we've learned to be patient with the ones who've come after us. Thank you for teaching us how to enjoy our companionship and to enjoy working with others and to see the excitement in their eyes as they grow into this really wonderful field that we've had. So congratulations to you for 50 years, and I know it'll be many more.